We are the team from Hong Kong. We are here to introduce our project, which is about biotech research with the usage of AI. First, let's talk about GENE. So, what is GENE? GENE is for generation. It means the spearheading next generation biotech R&D. And for our expression, our E, it would be expression, which means the transformation of abstract ideas into clearer images. And for our N, it would be networking, which drives engagements to raise awareness. And also for E, which is education, which means the empowerment of learners, which allows people to understand our own project. Our team has developed a novel AI-generated peptide drug that inhibits lung cancer. KRAS is a mutated master switch that drives lung cancer, but direct KRAS drug often fails due to rapid mutation. Hence, we aim to develop peptide drugs that block PDE delta, which activates KRAS to avoid resistance. Firstly, RF diffusion, protein MPNN, and dynamic bind are used to create and select free peptide sequences that can bind to the PDE delta. To improve its efficacy, a cell-penetrating peptide was added to binder 1 with a linker. At 50 micromolar, the fusion peptide led to an inhibition on cancer viability on A549 cells was approximately 80%.
We also studied the effect of the drug on tumor spheroid. 15 micromolar FPP1-CPP is administered and tumor viability was reduced by 20% after a 2-day treatment. Then, the effect of FPP1-CPP on a non-cancerous bronchial epithelial cell line was tested. Due to the favorable safety profile of the drug, it did not affect the healthy cells. In combination therapy, it is shown that FPP1-CPP can reduce the required dosage and side effects of cisplatin. Next, a plasmid is designed to express the drug inside the cancer cell. A549 cell transfected with the plasmid has a reduction with 40% of cancer cell viability. KRAS affects MAPK and ERK pathways, which promote CCND1 and MPAS genes. If KRAS is inhibited, these factors will also be downregulated.
We conducted qPCR to study the RNA expression under drug treatment. The annotation curve of CCND1 shows a higher CD value of 50 micromolar samples compared to the control. The right shows the fold change with reference to housekeeping genes. At the same time, the results on transcriptional target MPS show a significant drop in gene expression, suggesting that MAPK and ERK pathways are cut off by KRAS inhibition. Here are the references.
Next, we're going to talk about the 4 pillars of our research. I'm going to demonstrate the use of co-life AI in our research. For example, we use bioscene to predict the change of the A549 cell viability. Also, we've used RF diffusion to design the protein structures and generate the binding proteins or inhibitors. We also used biocircuit AI to design a plasmid-based genetic. We also used the biocircuit AI to design the plasmid-based genetic circuit to express the inhibitors selected in the cancer cell.
Let's talk about E, which is expression, which means the transformation of abstract ideas into clearer images. So for this aspect, we have used AlphaFold, which is a Nobel Prize-winning 3D visualization. We try to use 3D models in order to see how our peptide enters the binding pocket of our PDE delta, which is this silvery thing. And then so in here you can see that there is an AlphaFold. We have used AlphaFold in order to make this image. So you can see here, this is our PDE delta, the silvery thing. And the green one is our peptide, which is the drug that we are making. And you can see that in here, the peptide has entered into the binding pocket. And so you can see that it occupies the binding pocket. And this allows our KRAS to not being able to react with our PDE delta, and therefore which can induce cell death and also stop lung cancer.
Is networking. Promoting science can be challenging, but biotechnology shouldn't be limited to us in the lab. It should be for everyone. Students, gamers, people who've never read a research paper before. So we ask ourselves, how do we reach them? How do we make cancer biology fun? Our answer, Roblox. Millions of players, every day, all ages. We built Cancer Cells Sage, a wave-based shooter where you fight cancer cells. No boring lectures, no textbooks. Just shooting, dodging, and surviving 30 waves of enemies. Every boss fight teaches you something. Every enemy type represents a real cancer mutation, but you don't need a PhD to play. You just need to shoot. But building the game wasn't enough. We need to market it. That's where photo AI comes in. We used it to generate promotional graphics, AI videos that we can post and advertise on social media effectively. Here's the video.
Grab our Roblox game, Cancer Cells Siege. It's your real-world intel center with cancer research updates. Now lock and load. Let's find ways to smash cancer cells. Let's go!
The result, a complete package, a game on Roblox, marketing materials from photo AI, and a way to get biotech into the hands of millions. Science shouldn't sit in a journal. It should be played, shared, experienced. Because promoting science is not about dumbing things down. It's about meeting people where they already are. On to education. Spread the result of the investigation to more people around. To make our complex lung cancer research accessible, we created Poke ACPS. This card game maps our real wet lab data onto competitive mechanics, letting students use peptide cards to destroy cancer cells and learn biotechnology through interactive fun. To expand globally, we use Lovable to launch our digital hub. Building this web app with AI didn't just streamline our marketing outreach, it actually helped our team understand our own project variables and data structure much better. Finally, we utilized Factious AI to striking promotional poster. This assets instantly turned dense scientific statistics into clear visual marketing tools, providing that biotech communications wins when combining gaming with AI. Thank you for your listening.